So good afternoon students. So in the last class I told you about components of transdermal patch. With this I told you different components like liner, drug and PSA and permeation enhancers and microparticulate microporous or semi-permeable membranes. So with this backing film and pouching material. With this today we will discuss about types of transdermal patches. So generally these transdermal patches are three types: adhesive diffusion controlled, reservoir type and matrix type. Generally it's three types. So first of all we'll discuss about the first one, adhesive diffusion controlled. So this diffusion control means it is made up of, there is a single layer which contains the adhesive, and in that only the drug will be present. And this particular type of we can tell transdermal patches contains one simple liner and backing film which can be removed very easily, which is temporary, you can tell. That's only used to prevent the we can tell protection from the environmental hazards. And then coming to the second one, the adhesive diffusion control is classified once again two types: single layer drug in adhesive, multi-layer drug in adhesive. So in this single layer drug in adhesive and multi-layer drug in adhesive means in the single layer there is just only one layer of adhesive present, and in that only the drug will be entrapped, and in this from the adhesive layer only the drug will be permeated through the skin. So we can see here in the diagram, here we can see the upper part is the impermeable plastic membrane, drug impermeable plastic membrane, which we can tell it is your backing layer. We can tell this one is your backing layer. Then in the middle we can tell it's a reservoir of drugs which is directly connected to the adhesive layer. In the bottom part, this part is your adhesive layer. So from this adhesive layer the drug will be that drug is present in the reservoir will be transported to the skin through this adhesive layer. And that the backing layer, that is already I told you in the upper part, that is impermeable plastic membrane. Then coming to multi-layer drug in adhesive, multi-layer means two layers of the adhesive will be there, and in between there will be only one layer that will separate the two adhesive layers. So in these multi-layers it is like a simple single layer but two will be attached. When we are taking multiple drugs or we need to use it for a longer period, when one adhesive layer will be finished, another layer will be, from the another layer drug will be transported. In these cases we are taking multi-layer. So then coming to reservoir type of devices or transdermal patches, in this the first one membrane moderate system and micro reservoir system. The reservoir type, two types are there. One is membrane moderate system, another one micro reservoir. So in this one example I have given you here, you can see here in the top part it is your backing layer. We can tell it is made of we can tell it's your drug impermeable plastic laminate, drug impermeable plastic laminate, or simply we are calling it as backing layer. And in between that you can see small reservoirs of drug, and here you can see the adhesive layer, and here you can see the red permeable polymers, red controlling polymers which will control the release of drugs. In the bottom part we can see here the red controlling polymers from which the drug will be released. Here in the side we can see the adhesive layer which will attach the skin, and here the backing film will prevent from the environmental hazards, and from the reservoir the drug will transported through these holes present. Through these holes present it will transport to the skin. So this is your moderate membrane system. So like that micro reservoir system also there. In the micro reservoir system, there is very small reservoir system that will be directly attached to the skin. Directly attached to the skin, and from this micro reservoir drug will be transported. Then coming to third one, matrix type. So in the matrix type, generally we know that the drug will be dispersed in a layer, matrix layer, and through different types of mechanism, the drug will transport to the skins. Drug will come outside from this matrix layer. If you see the diagram here, this is the backing layer, we can tell, and in the middle, the absorbents are given, and here we can tell this one is your drug reservoir. This part is known as your drug reservoir, which will be in contact with the skin, and here in between the absorbent and drug reservoir there is another layer that will be attached to the that we are calling it as occlusive base plate. Occlusive base plate means it will selectively permeate small amount of drugs, and here in the side you can see adhesive tapes which will helpful for the attachment of the skin, and that will help to the use for the prolonged time. And from this drug reservoir only, the drug will be transported to the systemic circulation through the skin. So this is all about types of transdermal patches. Now we will discuss about evaluation of transdermal patches. So in the evaluation of transdermal patches, generally you can see here there are physical evaluation parameters there. First one is the physical evaluation. In this, first one is thickness. First we have to determine the thickness of the transdermal patch. Then percentage of flatness. What is the, how many means it is flat, how much it is flat, and how much it can be attached to the skin, percentage flatness. Then your folding endurance, third one. Then tensile strength is the fourth one, which is coming under physical evaluation. Then coming to another evaluation test, weight variation. So weight variation means batch to batch the same weight will be maintained for the total batches of transdermal patches. So that's why 10 we can tell patches, transdermal patches will be taken and will be measured the weight variation. Then from every next one, drug content, from the every transdermal patches drug content will be determined. Next one, moisture content. From there also moisture content will be determined by keeping it in desiccator and with some moisture absorbing substances, and we will find out how much amount of moisture it absorbs. Moisture uptake, how much moisture it uptakes, that is also one parameter. Then there is some evaluation parameter of the adhesive, means some adhesive that we are taking, using, how much it is efficient to evaluate that. There are some parameters, evaluation parameters also there, that already these parameters we have sometimes discussed in buccal drug delivery systems. In the buccal drug delivery system also, mucoadhesive drug delivery system, evaluation of adhesive materials also we have discussed. So let us slightly I will tell you about. First one is the evaluation of adhesive materials. We need to determine the peel adhesion properties, then your track properties, then your we can tell shear strength properties. Okay, these are the evaluation parameters of the adhesives. Next one, your confocal laser scanning microscopy. Confocal laser scanning microscopy. So this one shortly we are calling as CLSM. So we can know from this how much depthly it can able to penetrate the skin. So from this we can easily determine depth of the skin penetration of the patch, that we can determine by technique confocal laser scanning microscopy. Then also we are doing for skin permeation studies, how much amount of drug is permeated through the skin. For that there are some equipments we are using, instruments we are using. For example, membrane permeation cell and Jaward Lord cell rotating this system. Then your France diffusion cell, and like that so many types of instruments there from which we can determine the skin permeation study. France diffusion cell also popularly used for transdermal pairs. Okay, there is small equipment, small apparatus by which we can able to determine the permeation of drugs. So there will be two compartments in this France diffusion cell. One is receptor compartment, another one steerer, and in the top part we have to place the pads, transdermal pads, and slowly from the membrane, then we have to place one membrane there, then we have to see how much amount of drug is transported through the membrane and will reach the receptor compartment. So that will be later, sample will be collected and will be evaluated. Next one, evaluation parameter, skin permeation study. That also very important thing, skin permeation study. And next one, effect of skin uptake. Effect of skin uptake. How much amount of drug will be uptake by the skin? That study also have been done. And the next one, in vivo evaluation studies. In vivo evaluation study, sometimes we are going for animal models, human models, and different types of biophysical models. Okay. Then coming to last one, cutaneous toxicological evaluation. Okay, how it is safe on the skin? Is there any type of side effect on the skin or not? That will be determined. That is called as cutaneous toxicological evaluation. So in this first one, the skin irritability studies. Skin irritability studies means how much is it making irritation to the skin or not will be evaluated in the skin irritancy studies. Then your localized superficial infection studies means as it is attached more time, then is there any type of we can tell infection on the particular area or not, that can be also determined. So that is generally we can find out by knowing the growth of microorganism, microbiological test we can conduct, so that we can know the any type of growth of microorganisms are there or not. So these are all about your transdermal patches. So in the next class we will start another unit for you. Thank you.